Hello, I'm Chunyu Liu at SUNY Upstate Medical University at Syracuse. Um, I'm glad to be here to introduce PsychENCODE project and explain how PsychENCODE data might be useful for your study of psychiatric genetics. So PsychENCODE Consortium was established in 2014, funded by NIMH. Now it grows into a big consortium with 47 grants supporting 12 institutes, more than 100 scientists. So the consortium focuses on human brain. And we do have individual projects cover non-human primates, even mouse, but the majority of the study are on human. We cover several major psychiatric disorders, including schizophrenia, bipolar, autism, PTSD, major depression. But the big chunk of the brain sample actually from normal, psychiatrically normal people. We also cover a wide range of life stage from prenatal, fetal tissue, developmental, early life, and adult brain. So the major features of PsychENCODE, which make it different from all the other consortium like ENCODE and the GTACs, are the following 4 points, as I appreciate. First, genetic variation. We use population sample. We have hundreds, even thousands of brain acid sequence. So with that, we can study genetic variation for their contribution or regulational role in different omics. And also, we focus on brain, not other organ tissue. The major brain region we study from the cortex. We do have a few studies cover the other brain region, but frontal cortex is a major brain region for the time being. So the major disease, as I already said, schizophrenia, bipolar, autism, major depression, and PTSD. And we do have several different omics covered by different projects. For example, actually, I will have slides to show that specific information. In 2019, the consortium has the first wave of publication, 11 papers published in the Science family journals. And since the paper has been published, I will not spend time on the major results of those studies. And I just want to describe how you can access the data. So all the PsychENCODE produced data have been deposited into Synapse, which managed by Peter, Meta Peters, and Kelsey. So this is the website they created for as a public portal where you can browse the data and request access to all the data produced so far and released to the public domain. So there are already more than 200 terabytes of data produced by the early phases of PsychENCODE. As you can see here, the biggest data sets are actually RNA-seq data, then followed by ChIP-seq data and ATAC-seq data. So the other data are relatively small, like high-seq, normal-seq, and bisulfite sequencing for decomethylation. Subset sample have genotype data as well. Another web interface or a service or consortium is creating is this PsychScreen. This is a project led by Dr. Ziping Wen. Her group is creating a website for you to query PsychENCODE analytical results, including you can query the information by genes, by regulatory elements, by specific genetic variants. I think the genetic variant is a simple path. We will talk a little more about how they relate to PGC. So the major thing I want to deliver today is how PsychENCODE actually can be connected to PGC. So this blue box actually shows you SNP association with different disorders. Actually, the major products from PGC, those GWAS results. The green oval actually covers the major products from PsychENCODE. It contains all the brain omics for gene expression, for epigenomic measures, protein abundance. And with that, you can calculate QTL, including expression QTL, eQTL. That basically associates SNPs with gene expression. You can also do QTL for the other omics. And certainly you can do differential analysis, differential gene expression, for example. And other than that, you can also perform Mendelian randomization analysis, for example, to study the causal relationship between SNP and disease. So I want to spend a few minutes to talk about the project in my own lab. That is called Brain GWAS. It's one of the dozens of projects funded by PsychENCODE. So my first collaborators on this project is Kevin White at Chicago and Jun Min from St. Children's Hospital. My most recent project is collaborating with Stella from Mount Sinai and Iran from UCSD. We also have peripheral collaborators from Vanderbilt, University of Texas, and Central South University from China, and working on data mining, analyzing the data we have generated so far. I want to especially thank the program officer, Gita and Sander, who have provided us continued support. So from this Brain GWAS study, we have generated genotype data either by array or low-pass whole genome sequencing on more than 400 brains. Majority or most of them have been sequenced for transcriptome, RNA-seq. We also have a good proportion of samples have ATAC-seq, ribose-seq, and proteomics by mass spec. So through that, you can see this really covers the major components of central dogma, from genetic variants to gene expression, transcription, to translation. From translating the RNA bind to the ribosome, then translating to protein. So we use this multi-omics data along with genotype data. We can perform QTL mapping. We can also build a co-expression network. With those information, we can further extend to connect to the GWAS from PGC. So the goal is we want to use all the information first to understand and to explain the GWAS signals, also trying to capture some causal relationship, which is not obvious by just looking at the GWAS alone. And on top of that, we can also perform something like the TWAS analysis, so we can identify novel risk gene in the pathway and further build a prediction model. I want to share a few unpublished results. The first one is the QTL mapping of multi-omics data. This is a study done by former PhD student Jiang Yi. So he was analyzing our brain GWAS data, which contains, as I said, RNA-seq, riboseq, mass-spec, and ATAC-seq from 200 to 400 brains. So the result is it shows different QTL actually can produce different. I mean, different omics can give you different amount of QTL. This A panel shows you eQTL, splicing QTL, and actually you have many more eQTL and splicing QTL than ribosome QTL and protein QTL. And if you look at the effect size of different QTL, you can see eQTL has a stronger effect size than a ribosome QTL and protein QTL. I think that's easy to appreciate because gene expression, ribosome binding closer to genetic variants than the protein because it involves more steps in the central dogma. And they do explain a different level of heritability from the GWAS results as well. And when you compare the different types of QTL you obtained from different omics, you will see for those SNPs associated with the same gene pair at different omics, they have some good consistency. That is reflected at the positive correlation diagonal. But at the same time, you do see some interesting pair go into opposite directions. For example, in this panel, comparing eQTL with ribosome QTL, we do have a small number of SNP and gene pairs. They show a negative correlation. The same thing you can observe in comparing ribosome QTL to protein or gene expression to protein. At the same time, we realize there's still many gaps remain to be filled by the PsychENCODE consortium or the data analysis, particularly for the eQTL perspective. So we still did not cover well for the early development. We do not have good coverage for diverse population. We still need to cover in detailed cell type. We have several projects ongoing right now. This again, unpublished data, a collaboration between my lab and Michael Gandel at UCLA. This is slide prepared by Michael's student, Cindy Wen, showing the thing we are working on. We are generating eQTL, isoform QTL, splicing QTL on fetal brain tissue from nearly 700 sample, cover 3 different trimesters, 3 population source. And this data actually shows you the fetal brain do capture some unique early developmental QTL. And you can use those information to study, to analyze the GWAS signal and to see they're enriched in GWAS signal as well. as you observe in adult brain QTL. That's a study on fetal brain. We already have some cross-population study, but in adult brain, the situation is also there. So we don't have good coverage for diverse population. The current data we generate from bulk tissue RNA-seq predominantly European population. We only have an 18 African American brain, and that has been corrected to a degree in the single cell RNA-seq data. We have more representation from Hispanic population and African American relative to Caucasian sample. But so far, none of the non-European sample have been formally analyzed yet. So we have a project ongoing, also studying East Asian Han Chinese brain QTL. This is the project with my collaborators, Chen Cao in China. This slide prepared by our student, Chunyu, showing using 150 brain samples from China, we can identify also a big amount of, a large amount of eQTL and splicing QTL, referring to thousands of genes. So 80% of the E gene or splicing QTL gene actually are consistent across two populations. At the same time, it tells us that you still have substantial amount eQTL that only appear in one population than another. And the important thing is when we use the QTL to explain a GWAS signal, we see an interesting phenomena. So if you use East Asian QTL to explain East Asian GWAS signal, you have a higher proportion, significantly higher proportion can be explained. And then using European eQTL data. So I said we have a gap to fill about the cell type specific eQTL. So that is a major task for this current phase of PsychENCODE. Actually, right now, there's hundreds of brain is under investigation. I mean, is being sequenced for single cell or RNA-seq or ATAC-seq. We expect many major cell types will be covered. So stay tuned. In 2022, we expect there should be publication coming out from our consortium regarding cell type specific eQTL, early developmental stage QTL, and some population specific eQTL. So because of pandemic, this is our latest group picture. There's several labs joined the consortium later during the pandemic, so we don't have a group picture. As you can see, we have many seniors, investigators, and the young fellows in this picture. I believe everyone will have their own vision or interpretation of the PsychENCODE data, and you definitely can approach them and listen to their explanation or presentation about PsychENCODE. Hopefully, that will be a very useful resource for you to study psychiatric genetics. Thank you. Bye.